Hello everyone. Today we are going to discuss about the drug development. The drug development is the process of bringing a new pharmaceutical drug to the market once a lead compound has been discovered or identified through the process of the drug discovery. There are the stages of the drug development. It includes the first is a discovery and the development of the drug. Second one is a preclinical research. Third one is a clinical research. Fourth is a FDA drug review, and last one is a FDA post marketing drug safety monitoring. Let's see one by one. First is a discovery and development. In the drug discovery, research is how the new medication are the discovered. It takes place in the historically drug discovery, design and development mostly started with the identified the active ingredient from the traditional medicines or the pure by chance. Later in the upcoming eras or classical pharmacology was the use to investigate the chemical library, including small molecules, natural extraction, and the find those with the therapeutic effect. Since the human DNA was the sequence reverse pharmacology, where has found the remark to exist diseases through the modern testing. The step in the drug discovery are the screening hits, medicinal chemistry, and the optimization of the hits to reduce the potential drug side effect. The efficacy, potency, and the metabolic stability and the oral bioavailability are also improved in this step for the or step of the drug development process. First step is the target identification and the validation. In the target identification, it finds a gene or the protein where that can play the important role or significant role in the disease. After that, the scientist or researcher records the target's therapeutic characters, etc. The drug target must be efficacious, safe, enable, and capable of the meeting clinical and the commercial requirements. To validate the target, the researcher use the modern tools and the techniques such as the disease association, bioactive molecules, cell-based model, protein interaction, signaling pathway analysis, and in vitro genetic manipulation, antibodies, and the chemical genomes. The next step is a hit discovery process. In hit discovery process, assay development and the screening, high throughput screening, lead identification, and lead optimization takes place. In the assay development and the screening, the assay development in the drug discovery is a crucial component of the drug discovery workflow. The assay are the test system where they evaluate the effect of the new drug candidate at a cellular, molecular, and the biochemical levels. Next one is a high throughput screening. In this, it uses the robotics, data control software, liquid handling devices, sensitive detector to conduct or which are used for the millions of the pharmacological, chemical, and the genical tests, eliminating the hours of the painstaking testing by the scientist. Next one is a lead identification. In the lead identification, selection and the identification of the lead compound by using the various process takes place. The lead compound is a chemical compound that has a biological or a pharmacological activity for the therapeutic use. The lead compound identified by using a different type of processes in that serendipities, high throughput screening, random screening, and non-random screening, one of them. Next one is a lead optimization. In the lead optimization, the lead compound discovered and after synthesized and modified to it is improve for their potency and reduce the side effect of them. The lead optimization conduct the experiment by the testing by testing it on a animal for the conducting or collecting the data of the efficacy. Now next one is a preclinical research. Second one is preclinical research. In preclinical, once a lead compound is found, preclinical phases of the drug development begin in the in vivo research to determine the efficacy and the safety of the drug. The next one is in preclinical research. There it in that in vivo, in vitro, ex vivo. This type of the this type of a technique takes place in vivo. The preclinical research that are the development of the new drug using the mice, rats, and the dog model takes place. In vivo, in vitro, it is a research conducted in the laboratory, and in ex vivo, it uses the animal cells tissue from the non-living organism. Now next one is clinical drug development. Third, in that once a preclinical research is done or completed, researchers move to the clinical drug development, which includes the clinical trial and the volunteers study for or to furnish the drug for the human use, where the safety and efficacy of the drug is takes place. In the clinical trials, researchers studies that the test which are taken for the medical or surgical or the behavior intervention in the people. There are mainly four phases of the clinical trials. That is phase 1, which is for the trial to test the safety of the new treatment. Second, phase 2, where the trial tests a new treatment. Third, phase 3, which involves a large number of the patient involves, and fourth one is done after the drug has been approved. Let's see one by one. Before the 4 pages, there are a phase 0, which is also known as a microdosing, where the purpose of this phase is to help or speed up and streamline the drug approve process. At the same time, money spent in this would have been later phase. In this phase, minimum dose and the minimum volunteer is taking the participant. Next one is the phase 1. In phase 1, phase 1 are the designed to test the safety, side effect, based dose, and the formulation method for the drug. In phase 1, pharmacokinetic and the pharmacodynamic of the drug study takes place, so that the best and the safety dose can be found to discover the point at which the compound is too poisonous to administer. But next one, in the phase 1, phase is divided into two types, SAD and MAD. SAD means the single ascending dose, and MAD means the multiple ascending dose. In single ascending dose, small group of the substances given a single dose of the drug and observe for a period of time. And in the multiple ascending dose, a group of the subject receive the multiple low dose of the drug and observe for the period. Next phase is second phase, which is also known as a human exploratory. In that, it perform on the large group, that is 50 to 300, and it is designed to test or assay how well the drug works. It is conducted to evaluate the effectiveness and the safety of the new drug. Next phase is a phase 3, which is also known as a human confirmatory phase, in which these are designed to assess the effectiveness of the new intervention in which involves the large number of the peoples or patient are the involves where it is from the up to 300 to 3000 patient is volunteer patient is involves, which is found to determine the how the effect of the drug, how effectiveness of the drug is takes place, and compare the current gold standard treatment. Next one is last phase, that is phase 4, which is also known as a post marketing approval or a surveillance. In that the drug monitoring trials to assure the long term safety and the effectiveness of the drug. Thank you.